everyone, my name is Stephanie Singajaya and I'm going to present a paper with title How Were New Medicine Discovered? So, to start with, I will explain the introduction about why the writers raised this topic. The investment in drug research and development or R&D has increased, but the new medicines approved by the US Food and Drug Administration or FDA has not increased, and the attrition rates are very high. Without a dramatic improvement in R&D productivity, the pharmaceutical industry cannot sustain sufficient innovation to replace the loss of revenues because of the patent expiration for successful products. There are two important points in R&D productivity. The first one is efficiency and the second one is effectiveness. Efficiency means the ability to translate inputs such as ideas, investment, and effort into defined outputs, while effectiveness means the ability to produce outputs with certain intended and desired qualities. Since the dawn of the genomic era in 1990s, the main focus of drug discovery is drug targets, which are typically proteins that appear to have a key role in disease pathogenesis. There are many development of new tools to identify targets like RNA interference, compounds that interact with these targets, and even structure-based tools to identify and optimize such as X-ray crystallography. However, despite the power of these tools to identify potential drug candidates, R&D productivity remains a crucial challenge for pharmaceutical industry. Before the introduction of target-based approach, drug discovery was firstly discovered by phenotypic assay, often with limited knowledge of the molecular mechanism of disease. Nevertheless, the pharmaceutical industry's success to discover new medicines. Therefore, it concludes that the limited use of the phenotypic screening has contributed to the current lack of success in the drug R&D. In this slide, we will compare the advantages and disadvantages of target-based screening with phenotypic screening. For target-based screening, the advantages are have the ability to apply molecular and chemical knowledge to investigate specific molecular hypotheses, and the ability to apply both small molecule screening strategies and biologic-based approach, such as identifying monoclonal antibodies. And the disadvantages are the solution to the specific molecular hypothesis may not be relevant to the disease pathogenesis or provide a sufficient therapeutic index. While for the phenotypic screening, the advantages are this assay do not require prior understanding of the molecular mechanism of action or MMOA and can be translated more effectively into therapeutic impact in given disease state than in target-based assays. The disadvantages of this approach are the challenge of optimizing the molecular properties of candidate drugs without a design parameters that provided by MMOA, and the challenge to effectively incorporate the new screening technologies into the phenotypic screening approach, which is important for addressing the traditional limitation of these assays. So, the main purpose of this paper are to gain a better understanding of the factors that could contribute to the high attrition rates and to provide insight that might help to reduce this attrition and increase the R&D productivity. So, how they done the analysis? They will investigate the approach that were used in the discovery of medicines, analyze the characteristics of the new molecular entities or NME, and new therapeutic biologics that were approved by FDA during the 10-year period between 1999 and 2008 by examining the discovery approach, the MMOA, and whether the drug was first in its class. In the 10-year period between the 1999 and 2008, the FDA approved 183 small molecule drugs, 20 imaging agents, and 56 new therapeutic biologics. So it is 259 overalls. Out of this, 75 drugs were identified as first in class or with novel MMOAs based on the information provided in the product labels on the FDA website, and primary research also review publications. So in the discovery approach, there are 259 agents that divided into three general categories, such as first-in-class drugs, follower drugs, and imaging agents that were not further analyzed in this paper. So they just focused on first-in-class drugs and follower drugs. The method of discovery the new drugs are target-based approach, phenotypic-based approach, modification of natural substance, and biologics. All the biologics can be considered to have been discovered using a target-based approach. So why do they analyze the MMOA? MMOA is defined as the interaction between a drug and its target that creates a specific response, and this interaction function provides as therapeutically effective and safe response. The MMOA of the NMEs was analyzed because of three main points. The first one is the limitation of a target-based approach with respect to the MMOA that have been highlighted in some papers. Second one is the MMOA is a characteristic of drugs that has received less attention with regard to its connection to the attrition rates. And the last one, the MMOA can also differentiate similar drugs with respect to their therapeutic indication, for example, like aspirin with ibuprofen and naproxen. There are many aspects of this interaction that resulted in therapeutic outcome. For example, the site of interaction, molecular descriptor of the binding interaction, the functional impact, and the specificity of the functional outcome. The possible MMOAs at the target are: first one is kinetic mechanism, which is driven by a binding kinetics and residence time at the target, like equilibrium binding and also slow kinetics. Second one is conformational mechanism that involves a conformational change in the target as the response of drug binding, like non-competitive inhibition or antagonism, uncompetitive inhibition or antagonism, full agonism, partial agonism, and allosteric agonism. And the last one is redox mechanism, which is pharmacological response to the drug that caused by electron transfer between drug and psychological target. The MMOA can affect how efficiently a binding interaction is coupled to the functional response by considering its biochemical efficiency. The dose of a drug required to achieve the desired psychological response depends on its biochemical efficiency. Good biochemical efficiency enables efficacy at lower drug concentration and increase the therapeutic index. There are many factors that influence the shift in dose response curve between binding and functional assays, including the pharmacokinetics and ADME, assay relevance, the involvement of the target in the functional readout and biology, and the last one is the molecular mechanism of action or MMOA. So, the data in this paper will divide into three parts. The first one is the discovery of first-in-class medicines. The strategies that were used were categorized into four approaches. First one is phenotypic screening, second one is target-based screening, third one is synthetic natural substance, and the fourth one is biologics. First one, NME that were discovered through phenotypic screening. There are 28 first-in-class small molecule NME that were discovered in phenotypic screens. They came from the intentional targeting of specific phenotype or through serendipity. The intentional approach were based on the assays that measure a specific psychological phenomenon with little understanding of the MMOA. In many cases, the newly discovered molecules were later used to identify MMOAs for the psychological phenomena. The next one is NME that were developed as synthetic or modified version of natural substance or discovered by screening such substance. Only small fraction of the first-in-class NME, 5 out of 75, were developed as synthetic version of natural substance. Additionally, in some cases, natural substance provided starting points for small molecule in phenotypic screening. There are 10 NMEs here. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. And in target-based discovery, there are 3 NMEs. 1, 2, and 3. So, in total, there are 18 out of the 50 first-in-class small molecule NME originated from the natural substance. For target-based approach, there are 17 out of 50 first-in-class small molecules NMEs. There are various approaches contribute to these discoveries, and they are illustrated by these following examples. A target-directed screening of microbial growth from soil organisms resulted in the discovery of very potent, selective, and irreversible inhibitor of pancreatic lipases, which was named lipstatin. The development of 6 of the NMEs here that were discovered by target-based approach involve following identification of their effective MMOA at the target. These inhibitors represent success of the target-based strategy, but the optimal MMOA at the target may not be apparent at the time of initiating the discovery strategy. For the biologics that were approved under biologics license application and large peptide molecules that were approved as NMEs accounted for 25 out of 75 first-in-class medicines. The pharmacological action of these biologics included enzyme replication, augmenting existing pathway, providing a novel function, interfering with molecular activity, and delivering other compounds or proteins. Thus, the majority of these biologics function is interfering the molecular activity. The next one is the strategies according to the disease area. These tables show the discovery of first-in-class NME by therapeutic area. Evaluation of the discovery strategy by disease area showed that a phenotypic approach was the most successful for central nervous system disorder and infectious disease, whereas for target-based approach were most successful in cancer, infectious disease, and metabolic disease. And for biologics, accounted for most of the new medicines that act by modulating the immune system and 50% of the new medicine for cancer. For the second data is about the discovery of follower drug. There were 164 follower drugs, out of which 83 were discovered via target-based approach, 30 via phenotypic assay, and 31 were biologics. The third data is about the molecular mechanism of action. Nearly half, 22 out of 50 of the first-in-class small molecule drugs that were approved between 1999 and 2008 affected by the enzyme activity. The MMOAs of these drugs included reversible, irreversible, competitive, and non-competitive inhibition, blocking activation, and stabilizing the substrate. The next largest group of targets were receptors, most of which were G protein coupled receptors. Their MMOA included agonism, partial agonism, antagonism, and allosteric modulation. Next one, 4 of the drugs targeted the ion channel activity. Their MMOAs included the uncompetitive antagonism and partial agonism. And then 1 drug targeted the activity of a transporter, and the remaining drugs had other activities or still unclear targets or MMOAs. Here. Next part is discussion. The majority of small molecule first-in-class NME that were discovered between 1999 and 2008 were phenotypic screening here, which are 28 of the first-in-class NME that compared with only 17 from the target-based approach. At first, they conclude a lag time between the introduction of new technologies and strategies impact this trend, but such a lag is not strongly apparent in comparison of the cumulative number of NMEs here. Further analysis of the MMOA of the first-in-class NME also leads to propose that a focus on target-based drug discovery without accounting sufficiently for the MMOA of small molecule first-in-class medicines could be a technical reason that contributing the high attrition rates, because the MMOA is a key factor for the success for all approaches. There are 3 hypotheses that must be correct to result in new drugs. The first hypothesis, which is also applies to other discovery approach, not only target-based approach, is that the activity in the preclinical screens that are used to select a drug candidate will translate effectively into clinically meaningful activity in patients. The second hypothesis are the target that is selected must be important in human disease, and the MMOA of the drug candidates at the target is capable of achieving the desired biological response. In the case of phenotypic-based screening approach, assuming that a screening assay that translates effectively to human disease is available, a potential key advantage of this approach over the target-based approach is that there is no preconceived idea of the MMOA and target hypothesis. This could accelerate the identification of molecules with appropriate targets and MMOAs. However, there are two limitations of phenotypic-based screening approaches. The first one is that it will often be necessary to characterize the MMOA of active molecules that are identified in phenotypic screens to help the optimization of a drug candidate. The second one is phenotypic assays are often lower in throughput if we compare it to the standard target-based assay. For biologics and natural products, the discovery strategies that are based on natural substance have an inherent advantage. The biology, target, MMOA are often have been optimized already through the evolution. So, modifying such substance can be a fruitful approach. Similarly, some of the biologics that have been approved are utilizing endogenous mechanism in a rational way. For example, by providing a natural protein that is reduced in a certain disease, such as for enzyme replacement therapies for lysosomal storage disorder. In other cases, it is important to know the exact MMOA of biologics in their biological effects. The next part is follower drugs. With regard to the discovery of follower drugs, the opposite trend was seen compared to the first-in-class drugs, with the target-based approach accounting for 83 of this NME, and for the phenotypic-based approach accounting for 30 NME. This reversal of the trend might be resulted from the drug developers that taking advantage of knowledge from the previous identified MMOA and effectively use the target-based tools. A recent report also showed that research on a large percentage of follower drugs was initiated before first-in-class approval. Therefore, when a mechanism of action or a chemical class with potential to be developed into drug is discovered, multiple organizations within the pharmaceutical industry may pursue it, and this race may contribute to the escalating cost. Additionally, if a hypothesis that was later proven to be incorrect, the total cost could be higher. In the analysis, there are numerous diverse MMOAs for approved new first-in-class drugs, but drug discovery at present appears to be dominated by a one-size-fits-all approach, in which drugs are optimized for binding affinity with less consideration for binding kinetics and formations. For optimal application of target-based approach, it is very important to consider how efficiently binding is coupled to the response. So, for the conclusion, the analysis leads to conclude that the identification of an optimal MMOA has been a key factor contributing to the success of phenotypic screening in the discovery of the first-in-class NMEs in the 10-year period that was studied in this paper. Thus, the high attrition rates and low productivity in the pharmaceutical research and development could be decreased through the development and greater use of traditional phenotypic assay and by considering diverse MOAs when using a target-based approach. That's all about my presentation. Thank you for listening.